We are live with a with our virtual program called YY. That's WHY letter Y. So welcome to Willard Library. A closer look at DNA testing. I have two guests that will be talking tonight. The first is Jessica Brooks. She is our president of the Calhoun County Genealogical Society. We are having a joint program this evening with the Calhoun County Genealogical Society as a way to just get the word out about genealogy and tools that you can use to help with research. So I thought it would be fun to have Jessica with us. And we also have Ginger Ogilvy, which I had a lesson about how to say her last name. So thank you, Ginger. So welcome, Ginger, who will be doing the program for YY. So Jessica, do you want to say something before we get started?
Yeah, thanks, Melissa. I'm Jessica Brooks, and like Melissa said, I'm the president of the Calhoun County Genealogical Society. We've been in operation in Calhoun County for over 35 years, and the mission of our group is to support education and research and all things genealogy and family history for the people of Calhoun County and anybody who might have an interest in researching Calhoun County family members. We meet monthly from September through November, and then again from February through June. And those meetings are open to the public. Anybody can attend. Look for us on Facebook. Look for us in the shopper. Our email address is michigancalhoungene@yahoo.com. Gene is spelled Gene. We'd love to have any of you join us at our meetings. If you'd like to become a member, dues are $15 per year, and there are some benefits that come with that. So we're really happy to be with you tonight, and we're very thankful to Willard Library for co-hosting this event with us and helping us bring Ginger back to the society. I'm just going to say a couple things about Ginger, if that's okay. So Ginger is a professional genealogist and founding co-administrator of the Ogilvy Y-DNA Surname Project through Family Tree DNA. And as far as our society goes, Ginger has already been a presenter for us. You might remember her from last year if you tuned in to our January and February DNA series. Ginger has come back to keep teaching us about DNA, and we're really excited to hear what she has to say. Thanks so much, Ginger.
Okay, are we ready to get started? Awesome. Yeah, thank you so much for the opportunity to come back and talk a little bit more about DNA. I'm excited to share. Honestly, I get the most excited about Y-DNA. There's just it's just a really, really cool tool to work with. One of the many tools that we can add to our genealogical toolbox. Before I get started, I just want to say a couple of things that I always say when I talk about DNA. One is that what I'm presenting is current as of today, but the world of DNA changes very, very quickly. So if you want to stay updated on the latest and greatest, be sure to consult the resources that are in the handout, which hopefully you've been able to access. So, you know, just take it for what it is now, but remember that things can change, and things have changed just with Y-DNA over the past, I guess, 9 or so years that I've been working with it. So stay tuned for more developments in the world of DNA. The other thing I always say is that I'm as much as passionate as I am about working with DNA, I'm not a genetic genealogist. There are people out there who know so much more than I do. So my goal in bringing this program to you tonight is to just give you a taste, kind of take those first couple of steps forward. And for people who are maybe more familiar with other types of DNA tests, and you're thinking about Y-DNA, but aren't really sure yet, I just wanted to give you kind of a peek or a window into what the test results look like, because it's a little different. So not being a genetic genealogist, I definitely once again want to point you to the resources that are in the handout and that I'll be referencing during our program. So with that, we will get started and talking about why, what are the reasons why we might think about this type of testing. So also what I always talk about with DNA programs is this concept of both our genealogical inheritance on the one hand and our genetic inheritance on the other. So, for some people, those are exactly the same. But for other people, we may have been raised with stories or ideas, photographs or written records that we have received from parents or grandparents, great aunts, or what have you. We have this story that's been handed down from generation to generation about who we are and where we come from. At the same time, within our bodies, we have a story that also sort of unfolds to help us understand who our parents and grandparents and, going back many generations in some cases. So in some cases, those two stories are exactly the same. And in other cases, you may end up having two stories, the story you grew up with, and then the story that you're learning from DNA research. So keep that in mind moving forward. Tonight, I'm going to be talking about my Ogilvy line. I kept my maiden name when I got married because I love my last name so much. I grew up hearing my mom explaining to people how to spell it over the phone, and we were very certain that it was spelled with an IE and not a Y. Or all the other, I think over time I've collected probably around 25 variants of the spelling of this last name. It's a doozy to spell. I had to spell it for someone over the phone just today. But it's pronounced Ogilvy. And what I've learned, if you go out and about into the world, is that it's a name of Scottish origin. It comes from a very specific region of Scotland. It's sort of like the gateway to the Highlands from the southeastern side of Scotland, north of Edinburgh, and actually just outside of Dundee, if you're familiar with Scotland. My specific heritage, that's my dad farthest on the left, and then his father, my paternal grandfather, and going back all the way to, you can see the headstone there. That's my furthest, I guess I can go one more generation past that. But the headstone there is for a man named John Ogilvy, who the story goes that he was a loyalist. He came from Georgia. And during the American Revolution, he had a brother named Peter, were both invited to leave the new nation of the United States of America. They were refugees in East Florida. So they left Georgia and went to East Florida, which is basically like Jacksonville, Florida. And then were put on a boat and sent up to Nova Scotia. And so I can easily trace my line all the way back. I've actually visited that headstone in Nova Scotia. John, you probably can't see it, but John was 105 years old, apparently, when he died. His son, George Byers Ogilvy, is the next one in the black and white photo. And the gentleman after him is also named George. Those two, the father and the son, were pioneers who left Nova Scotia and settled in Utah. And that was in about 1855. And my family has been in Utah ever since. So I can trace my line all the way back, and I've got a map here for you. So I can follow my line from Utah back to Nova Scotia. And from Nova Scotia, we have Loyalist records that bring us back to Georgia. And then from Georgia, we start running into a big question mark. So for a long time, most of my growing up years, everyone thought that John, the headstone guy, he and Peter, their father's name was James. And James Ogleby apparently is how it was pronounced. We don't know where he was born, but everybody tried to make him come directly from Scotland. But over time, as I've dug into the migration patterns of people who were of Scottish origin, who settled in that region, particularly the region of Georgia that he settled in, they may actually have come from Northern Ireland. They may have been Scots-Irish. But that said, there were people with this surname who came from England. And of course, they could have come directly from Scotland. So that is still an unknown question. And we know that eventually we get back to the very early origins of the surname in that very specific region of Scotland. So that's the story. That's my genealogical inheritance that's been up to Georgia, has been verified with historic records. So why would we think about Y-DNA in this case? I forgot to count the number of generations there, but I think we're talking about 7 or 8 generations. With autosomal DNA, that's the DNA you may be more familiar with. That DNA tends to drop off around 5 generations or so. You could maybe stretch it to 7 if you're lucky in terms of finding matches, but that's about it. It really peters out earlier than 7 generations, but definitely by 7 generations, you're kind of out of luck. But the beauty of Y-DNA is that it picks up right around 8 generations. It's not very, very specific, but it goes back a little bit further. And so at least for the timing of my family and our generations, it picks up very nicely. And I think for other people who may have ancestry from colonial America, that is where we start to wonder, how did people get across the pond? Where did they come from? And so it really nicely picks up right there. The beauty of Y-DNA is that it can help you go really far back into your distant origins. And I'll explain a little bit more about that coming up. You can definitely use it to confirm or refute family myths when it's applied to the right situation, the right questions. You can use it to find your distant cousins, find other descendants of the same paternal ancestor. It can also help you figure out if potentially there may have been adoption or, just unexpected parentage, unanticipated changes in your paternal line. It's definitely very helpful. And I was talking before we started, it definitely came in handy to help me understand a case that I worked on recently that we used Y-DNA to help us understand and explain certain outcomes. And then when it comes to forensic investigations, whether it's a law enforcement case or whether it's identifying military remains, it can definitely play a strong role there as well. So quick overview. I don't want to spend too long here. I've talked about this in the past, but just to remember, I've enlisted that's my baby grandpa there, and his father, who was another George. He was William George. Lots of Georges along that paternal line. So they are here to help us kind of understand the different types of DNA testing available. So for autosomal DNA, you inherit it equally from both parents. You get the same amount of DNA. So 50% from one parent and 50% from the other. And anyone can take an autosomal DNA test. For mitochondrial DNA, that's just for your maternal line, mother's 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 mother. Men and women inherit the mitochondrial DNA equally. And so anyone can test, but it will only tell you about your maternal line. And then when it comes to Y DNA, it's only for your paternal line, your father's 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 father, going way back. Only males that have inherited a Y chromosome can take this test or will have valid results for this test. We'll talk a little bit more about that. So it's only for, if you want to know about your paternal line and you're female, then you're going to need to enlist your male relatives to help out. So just to kind of help you visualize this with the autosomal DNA test, it covers your entire ancestry, but it only lasts for so many generations before you start to lose, before you no longer have very good matches with cousins to help you understand who those further back relatives are. With mitochondrial DNA, it doesn't change. The mitochondrial DNA does not mutate very quickly at all. So it goes really far back. But that means you're going to have lots and lots and lots of matches with people who do not share close ancestry with you. But it can really help you if you're trying to figure out which region of the world your maternal line came from. And similarly with the Y-DNA test, it does mutate a little bit faster than the mitochondria, but it, again, will only show you the male line and also goes pretty far back, much further back than your autosomal DNA. So just to help you visualize, you're probably familiar with a pedigree chart. We're only talking about the very top line of your pedigree chart. Everybody else below this type of testing will not tell you anything about the rest of those folks unless you have, multiple lines converging, going back. But for most people, this will be just only your paternal line. So to help you, kind of picture this, if I wanted to know about my maternal grandfather, you can see there, that's George Virgil Russell, my mother's father. If I wanted to know about his Y-DNA, then I would need to get my paternal uncle to take a test. And then that would tell me only about that Russell line, only my mom's paternal line there. So you have to be very strategic about who is testing in order to answer the question. So make sure you've got the right tester to answer the question that you're interested in. And just to help, it took me a while to kind of figure this out. So I thought I'd put this in here just in case people are wondering, what are we testing? In the cell, we have the mitochondria here, and that's sort of the energy production of the cell. And this is, that's where we're getting that maternal information from. For autosomal DNA and for Y-DNA, those are coming from the nucleus of the cell. And so you may see the term nuclear DNA bandied about if you're out there doing research on the internet. And so all that means is nuclear just means it's coming from the nucleus of the cell. So let's take a closer look at that. For humans, we have 46 chromosomes. We group those into 23 pairs. The first 22 pairs are called autosomes. So they're bundled together, and you can see that here they're lined up from longest to shortest. And then the very last pair at the end, you can see are the sex chromosomes. So in this case, this is for a genetic male. They have one X and they have a Y. And that's that little tiny guy, the very last one right there is the Y chromosome. And that's what's being tested for Y-DNA. And this is, I don't want to lose you here. I know this looks really technical, but the part on the left in blue, I just wanted to, for those of you who are interested, give you a sense of what is it that we're actually testing. So we have that tiny little Y chromosome, and then in that tiny little chromosome, we're only testing a small part of it. We're not even looking at the entire thing, and that's the U chromatin right there that's in the bracket. So this is the part I want you to hold on to as we move forward, and I will I will be referencing back to this as we go. But I want you to think about Y DNA in two ways. When we have testing, the results that you're going to receive are two types of results. So the first one is what we call STRs. So we have STR testing, and STR stands for short tandem repeats. The way I remember this is that STR, I can kind of picture that word stutter. So if you think about, if you're stuttering, you're sort of repeating the same thing over and over. Or I don't know, for some reason, I always think about like a mimeograph. For those of you who are old enough, and a mimeograph machine or a copy machine, sometimes it grabs too many papers or it prints too many all at once. It's not a problem that this is happening, but basically when we submit that Y-DNA sample, we're going in and identifying how many times we have a repeat. And so here I have highlighted 3 testers. And if you'll remember back to what you know about DNA, that we have these base pairs that go together. And this is where we're looking at repeats going down that strand of DNA. So the first tester, you can see it goes ACT, 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 ACT. So that's 4 times that ACT is repeated. So we're giving them the value of 4 for that one location. For tester 2, you can see that there's an extra repeat there. It repeats 5 times. So they will be given that numeric value of 5. And then tester 3, we're back to 4 repeats. So the thinking here is that when you would have tester 1 and tester 3 have more similar, a more similar situation than tester 2. Theirs is a little bit different. So we're going to be honing in on what's similar and what's different when we compare different testers. Okay, so that's STRs. And that's what we're looking for with our Y-DNA testing. Another thing that we find with Y-DNA testing, and actually also with autosomal testing, are SNPs. That's SNP, and that stands for single nucleotide polymorphisms, which is a very long word. It just basically means it's kind of like a typo, where there's a change or a mutation in a location where other people have something else. This person, you can see tester 3 here, has a G instead of a C. And so over time and with enough testers, you can start to group people who have the same SNPs, the same changes in the same locations. And so those are the 2, there's STRs and there's SNPs. So we'll hang on to that. And that's also in the handout for you to reference later if you forget. So again, the short tandem repeat is a repetition of base pairs at very specific chromosome markers. And I'll show you how that plays out with testing. With the SNPs, the single nucleotide polymorphisms, that's a variation in a single base pair in a DNA sequence. And that's where we're thinking about a typo. There's just one change. When you have enough people, when you have a group of people who share a common ancestor, they will have the same DNA variations. They have similarities. And that's where we start thinking about haplotypes. That's, or in other words, a genetic signature. Something that says these people are similar and they are different from the rest of the people. And if you get enough of those people on a larger scale, then we start thinking about haplogroups. So that's basically a group of many different people with their haplotypes that come together in a large group. So here's a map to just kind of get you thinking about haplogroups. Again, these are larger sort of generalizations, I guess you could think about it. But I like to think about it as it's grouping people almost on a continental level. So you can see like over in the Americas, we have Q3 that extends actually through much of North America and also much of South America. If you go over to Africa, you can see there are different haplogroups there, but they all start with E, or several of them start with E. So, there's some similarities there. Or if you look over at Australia, not Australia. Yes, Australia. I was thinking Austria, Australia. And this also, as people have studied these haplogroups, they've been able to trace migrations over time. So starting in Africa and then flowing through the Middle East and then kind of branching off into parts of Asia or parts of Europe and then on over to the Americas, as you can see. So people have questions. This is what we call deep genealogy, very, very thousands and thousands of years going back. Trying to understand our origins and how the flow of humanity has taken place over time. It's kind of exciting stuff to think about. Okay, so if you're interested in participating in Y-DNA, there's basically just one place to go. I'm not just saying that, like this is the place to go if you're in the US for Y-DNA testing. And that is called Family Tree DNA. You can also do other types of testing there. You can do mitochondrial testing as well as the more typical autosomal testing. The tests are quite expensive, I will say that. One strategy that you can use is to wait for sales. There are certain times of the year, like generally Father's Day or Black Friday, November-ish, kind of leading into the holidays. You'll see some tests starting. Some sales starting for the tests. But I also wanted to point out here that there are different levels of testing. So what's considered a good start is, you can see that's Y37, and that's the number refers to how many locations, how many STR locations are we taking a look at. So we have Y37, and for a lot of people, if your funds are limited, you can start there, and then you can always upgrade over time. And so some people, like we did that in my family, we started with 37, and then a long time ago, they used to have a Y67, but that's not available anymore. But that was our next step, and we probably waited about a year to go up to 67. You don't need to submit a new sample every time you upgrade. They will hold on to your sample and then do the additional testing if you upgrade. So then the next step up at this point would be the Y111. And then from there, it goes to big Y. So it used to be big Y 500, and now it's big Y 700. If funds are not limited at all, then I usually recommend that people jump right in and go for the big Y, go for that big Y 700 and just get it done. But if you want to pace yourself and, kind of take advantage of those holidays or, birthday presents or things like that, you can kind of take the slow route. Okay, so I always use this slide for just reminding us that with DNA and really all of our tools that are available, we will be so much more efficient and get to the answers that we're looking for if we have a well-developed research question. So you can take a test and just kind of see what happens, but sometimes you want to step back and think about why do I want to do this test? What is the question that I'm trying to answer? And how can I be strategic? That definitely applies with Y-DNA testing, that the strategic testing where you identify specific people to help you answer questions is going to be really important with the Y-DNA. So for example, like I said before, if I wanted to know more about my mom's paternal line, then I wouldn't ask my brother to test, because that will only tell me about my father's paternal line. So you want to, just keep those things in mind. So my question is, and really the question of many Ogilvies, is where did we come from? And in order to get to that place, the first step we needed to do was start developing a haplotype or a genetic signature for understanding our specific line of Ogilvies. So you can see at the top, I have James Ogilvie. He had two sons, John and Peter, who were approximately 2 years apart. So you can see that's the headstone I mentioned there, John Ogilvie. Born in 1751, and my father is a direct descendant of John. And then I was able to go over to Peter's line and identify a direct descendant of Peter. And I found this individual, and their family had been, also seeking the answers to the same questions that we were. So they were very happy to participate with us. So this living direct descendant of Peter is an exact, straight across the board, sixth cousin with my dad. So the first question was, if my dad and this cousin take a Y-DNA test, will they show up as matches? So that's a lot of generations and a lot of marriages where we have to trust that the information, the genealogical inheritance that we have is backed up with our genetic inheritance. Okay. So we went ahead and had both of them test for Y-DNA testing. So you may be familiar with triangulation for autosomal DNA. It's a little bit different for Y-DNA. For autosomal DNA, we are thinking more in terms of almost like a pyramid with your most recent ancestor at the top, and then you're looking for at least 3 testers who all match each other. But for Y-DNA, you have the tester and you have their match. And then if those two match, then you know that they go back and they connect at the what we call the most recent common ancestor. So you just need to find that one match, and then you need to do the research to find out where your two lines come together. So okay, so we took a test, and I'm going to give you a few screenshots to just help you understand what it looks like once you get in to Family Tree DNA. So we have, I've got an arrow there pointing to the section that's just focused on Y-DNA. You can see you have an option to look at your matches. It tells you what testing level you're at. So this screenshot is actually older. This is from when my dad was at Y111. He hadn't quite taken the big Y test yet. You can see that there's a haplotree there, and I'll give you some examples of that coming up. If you look on the top right there, you can see the group projects that my dad has joined. So we ended up actually starting, I worked with another Ogilvie person who's not super closely related to my dad, but did show up as a match. We worked to get the Ogilvie surname group started. And so you can see that's one of the group projects there. And then there's some other group projects that my dad joined, including one that's just generally Scottish DNA. You can also see on the right that it says predicted Y-DNA haplogroup, and that at the time was R-M269. That is a very general haplogroup for men of Western European heritage. So it didn't tell us very much. And the reason why that was just predicted at that point is because my dad hadn't taken the big Y test yet. So all the tests up to up to the Y111 will give you only STR results. But when you go for the big Y, that's why it's called big Y, is you get the STR up to 700, and then you also get SNPs. And the SNPs are what help you understand what your haplogroup is. Okay, so that's what the sort of homepage looks like. And then once my dad took the big Y test, you can see there's Y-DNA tools still there, but a little bit below there is the big Y tools. And that's where we start getting into what's called the block tree. There are some haplogroup reports there. The big Y matches is more based on SNPs than the STRs that you'll see in the regular Y-DNA results. You can click on your YSTR results there. There's an option there for just looking at basically, I'll get into this, but the string of numbers that you get as your YSTR returns. So if you click on your Y-DNA matches, then you can see who is showing up there. So I've gone ahead and erased the names of the matches there. So if you see that blank column on the left, that is where you'll see the names. And people often will get to this point and they'll start seeing people who may have the same surname as your tester. But you may also start seeing a lot of other surnames that you're not familiar with. And I want to exercise, I put this in the handout too, but you exercise caution here, because sometimes people will look at that and they will start to kind of worry that there may be infidelity, there may be, maybe not the parent expected going on. But these matches tend to go back very far, hundreds of years in some cases. And a lot can happen during that time. And we have all sorts of situations. There may be adoption. There may be, like in our case in Scotland, we have clans where people may share a surname, but they may not be biologically related. And then, of course, don't forget, if you come from a place that uses patronymics, like in Scandinavia, where like I have Jens Andersen, who is the son of Anders Jonsson, it's that surname is not going to be repeated generation after generation. So there may be, some of that going on. So just, don't get too worried if you see other surnames there. I want to call your attention here to the column headings. It will tell you for each match how high their testing level is. It will tell you the genetic distance. It will tell you if they participated in Big Y, and if there are, what the Big Y results are there for STRs. If they know the haplogroup, they will tell you, or they'll tell you the estimated predicted haplogroup. They may tell you the paternal country of origin. So you can see I was pretty excited to see Scotland showing up here. And then the column that says paternal earliest known ancestor, this is self-reported by the tester. So just be aware that there may be some errors there, or there may be some predictions going on there. So it's not necessarily set and it's not verified by any outside party. And the next thing I want to bring your attention to is you have the option to download your list of matches. That export CSV option to the right can be very useful for your own records. And then to the right of each match, you have a family tree pedigree option. You have a place for notes, And then you also have a time predictor tool that I'll show you what that is coming up. I also want to show you I want you to note here at the top that you can actually toggle between the different levels of testing. So to show you how that works, it's sort of like a microscope, right? Where you can shift the lens and you can dial it in. You can adjust your focus on what you're looking at. So just to highlight here, if you start at 12 markers, you can see in this case, there's over 1,000 matches showing up there at 12 markers. That's not going to show you very much. They may be very, very, very distantly related to you, but the information that they have will most likely not be very, very helpful for you. What you want to see are more the higher testing individuals. However, you start to lose people. Some people haven't upgraded. Some people, So basically you can see how it really drops. When you go to 25 markers, you're only at 81 matches. At 37 markers, you're at 14. And then by 111, you've only got six matches. So I'll talk a little bit more about that, but I just wanted to highlight that's an option for changing how you look at your matches. And then I mentioned the time predictor tool that's over on the right. I've got that circled there. But so if you click on that on the right, it will take you to this time predictor. And so you can do this with each of your matches. You click on that and it will give you this sort of report. It used to be called the tip report. So in this case, I clicked on someone, and I'm not sure if you can see this, but on the far right, under Y111, it is highlighted in blue, and it says 1650 CE, which is the Common Era. And then at the very bottom, it's basically saying that this match has a genetic distance of 5 at the Y111 test level. And based on that, my dad and his match are estimated to share a common paternal line ancestor who was with a 95% probability born between 1450 and 1800 CE. So you can see that's why 1650 is sort of in the middle of that. That's why that's highlighted. The most likely year is rounded to 1650. So it kind of gives you a sense. You're like, well, I have this match, but maybe I don't understand what genetic distance means, or I'm not really clear. It gives you this kind of report on how closely you and this match would be. So you can see 1650 is pretty far back. And I also want you to note that you get more information, more specific information at the higher level of testing. So if I looked at this same match, I would still, I guess it looks like it has that 1650 option there for Y37. But the higher you test and the higher your match tests, then the more you can drill down on where your lines diverged. So I'm going to show you, I mentioned that string of numbers. I just want you to first, before we go into that, understand really quickly that the darker blue is so these are all, we're talking about locations on the Y chromosome, where we are at each location, we are looking for that STR result. So remember, that's the stutter. That's where we're looking for repeats, and we're getting a numerical value for how many times we get a repeat there. So the further to the left, the first 12 markers are have a dark blue heading, and then those headings lighten up as we go. So like Y111 is the lighter blue, and then intermixed in there, I'll show you in a second. But wherever you see that sort of rust color, those are faster changing STR markers. There are certain locations that tend to mutate more quickly than others. So this is from our surname project. And I love being able to look at this. It's just so fun. So on the left, you can see there's a list of different testers. These are all people who are participating in the surname group. And by the way, this is publicly available. So this is just a screenshot from our publicly available surname project. And I've highlighted my dad is the first one here in the yellow, this yellow line. This is his very unique to him string of numbers that are the results, his STR results. And so you can see we have 12 and then moving into 37. And then way over here, we start to get into 111. And then I've highlighted down here, this individual here is my dad's cousin. This is the 6th cousin who tested. And you can see here that they have similar numbers going across. So that's kind of what we're going for. We were really excited to see him show up as a match. So just to explain this just a little bit more, we'll kind of drill down a little bit more here. But if you look at each of these columns and follow it down, just pick a random column that you can see and look at the numbers following it down and see how you can get different numbers. So for example, over here, I don't know if you can see this with my cursor, but at DYS 439, it's the rust colored column more to the right. We have a 10, and then we have 2 12s, and then we have an 11, and then we have 2 12s again. So the people with the 12s are going to be more closely connected than the people with the 10 or the 11. So you're looking for people who have similar numbers all the way across. And again, these numbers that you're seeing are the numeric values that come from how many repeats are at that location. And so we're looking at, you can see this is within our surname project. We're seeing a lot of similar numbers coming down the columns. But for example, here under DYS 458, you see 3 17s that are highlighted that are different than the 16s above. So we might start wondering perhaps those 3 individuals might be more closely related to each other than they are to the preceding people with the value of 16. And we start to look at each column individually, but also look at all the columns. And anytime we start to see a difference, so again, like that person who has a 17 instead of a 16, that is what we call a genetic distance. That is one difference. And so every time you have a difference, that's going to be another number that's added to your genetic distance. So you want to have a very low genetic distance when you're comparing your test to someone else's, if you're looking for people who are closely related. Another thing I want to highlight here when we look at, so this is back looking at our match list, you can see that different people have tested at different levels. We've got a big Y 700 at the top, big Y 500. Then we've got a couple of Y 111s, and then we're back to big Y 700. So that is going to make a difference. So we want to understand that some of the matches are not going to show up at those higher markers just because they haven't tested at that level yet. They maybe, like my family, started out at 37. They haven't made the upgrade up to 111 yet. And so they may just not show up. It's not that they're not showing up because they're not closely related. It's just that maybe they haven't upgraded yet. So that would be a strategic person that if you're interested in them, you might want to reach out and say, hey, have you considered upgrading? It's kind of funny. It ends up kind of being a little bit of an arms race. Like with my dad's cousin, we started at a certain level and then they would meet us at that level and then we would upgrade and then we'd ask them to upgrade. And so, you've got to pay attention to what level, not only what level you've tested at, but also what level your matches are testing at. Okay, so I think it's on the second page, maybe the third page of your handout. There's a blog post that talks about this, and I was so excited I had to try it for myself. I hope this makes sense. I really wish someone had laid this out for me when we first started. So sometimes people aren't showing up because they haven't upgraded, but sometimes they're there and then they drop off and then they show up again. And it's just because of how many, what the genetic distance is that may or may not be factoring in terms of the threshold that family tree DNA is using. So in this case, looking at this, I just picked my dad's top matches. And I looked at how they're matching at 25, 37, 67, and 111 levels. Okay. So I started out with ordering them all the exact matches at 25 come first, and then one genetic distance, one step away are here. And then we go to 2 steps. And then if you'll notice, there's a couple people here that aren't showing up at 25. So pay attention to those as we go. And I want to explain this because a lot of times people will say, well, we tested at 37. We're at Y37 and we hardly have any matches, or we have no matches. I've heard of people having no matches. And they're like, well, I'm not really going to upgrade until I have a match to match with, which definitely makes sense, right? But because of the way that this works, it's not necessarily true. And so this is what I'm going to show you. If I can get my cursor in the right place. Okay, so when we shift our lens, remember that microscope I showed you? If we shift the way that we're looking at it and we look at 37, the level 37 matches, you can see that the exact matches here shift to 2 steps. Here, for the one genetic distance of 1, you've got one person who dropped really far from 1 to 4. And then further down, you can see that people at 2 steps are really variable. One stays at 2, one drops down to 3, one drops to 4, and then 2 drop out completely. And then we have this guy over here that just shows up out of the blue. He's late to the party, but he's there. So, and then when we shift again to look at 67, you can see how these exact matches start to be pretty variable. The one that shifted down to 4 is now at 6. We've got a couple here that have dropped out, but note here, and I circled this here, these individuals that drop out when we shift to 67, if you check, they only tested to 37. So that is the reason that they're not showing up as matches at 67, okay? Same thing with this one down here at the 2 steps. But then also, here's this other guy showing up, wasn't there at 37. He dropped off, but now he's back and he's got 6 genetic distance of 6, which is the same as this individual up here who started out at 1. So you can see that there's some variability, and there's people who are dropping off for various reasons, and then people who are showing up. And then when we shift to look at 111, then out of the blue comes this individual down here who wasn't there at all. They weren't there at 25, and they weren't there at 37, and they weren't there at 67. And now all of a sudden, here they are at 9 steps, which is closer than this individual who started out at 1 step, and now they're at 10. So I just wanted to note this, that, you know, if you think that you shouldn't upgrade from 37 because you don't have many matches, that is, it's logical, but it's still a good idea to think about upgrading, because there might be people who are showing up in those other areas. So just wanted to be sure to highlight that. Okay, so that's enough about STRs for a minute. For SNPs, and I don't want to belabor this, but I think it's important to think about haplogroups, because this trips people up. Over on the right, you can see that this is from our surname group, surname project. Where it's red, that is predicted. So remember, that's what my dad's predicted haplogroup was R-M269. Okay, and then in green, those are haplogroups, and that's from testing what's called the terminal SNP. So like following it all the way through to the end, and saying this, you know, if you think about a tree, you know, and you have the trunk, and then you have main branches, and then you have tinier branches, and then eventually you get to the very outer leaf. That's what that terminal SNP testing is doing, that you get from the big Y. And that's helping us to really drill down and separate individuals or group individuals. And they have, I don't know about you all, but I have a really hard time when numbers and letters get mixed together. Like, it drives me nuts, like cards having numbers and letters mixed together. It just, I don't know what it does to my brain, but I have a hard time. So this, like that R-BY160432, my brain is like, so it helped me to be able to look at the haplo tree that's available as a tool on the website. It also helped me to do a quick search. So if you are getting this haplogroup information, go ahead and just search it. You can search it on Wikipedia, or just plug it into your favorite search engine. And this is where I learned a little bit more about what it means to have R-M269, which is basically, you can see on the map, it's broadly Western European. It's not very specific, but it is, you know, on a continental level. So here's the haplotree that is being pointed to here. You can click on that, and then it will take you to just like a series of drop-down boxes. And again, think of it like a tree, sort of unfolding from a branch to a twig to a leaf. So I was able to identify R-M269 here. It's highlighted a little towards the bottom with that gray stripe, and it fits under what is known as haplogroup R, which has another name there, R-M207. And so I'm going to walk you through, like if I were just following the drop-down boxes all the way through. Also wanted to note that you can look at the countries of origin self-reported from your fellow testers who are showing up in these different haplogroups. So that's the countries option. And then you can also, instead of having the countries there, you can also take a look at different SNPs. So if we follow R-M269, that's the predicted haplogroup, and we take a look at what some of the matches are showing up with their haplogroups, we can keep following it down. And here is this R-S7174, which is Ogilvy's. So my co-administrator was really excited to get that SNP basically named for the Ogilvy family. So that was pretty exciting. And basically, I just took all those boxes that you just saw and lined them up here to kind of give you a sense of how they all fit together, sort of like nesting bowls or measuring cups, how they all fit inside of each other. So I wanted to do this because you can see how I have these people in a haplogroup, and like you can see that R-S7174 is showing up. And I was curious about how that connected to the other one, which is R-BY160432, right? The head exploding one. And you can see how if you follow it through the haplotree, that one just fits inside of the other. It's just sort of downstream. It's just a little further out along the branch. And so if you're seeing two different haplogroups that don't seem connected at all, make sure you check them on the haplotree, because you can see that they may actually be very closely related or connected. And so don't let that throw you if you're seeing different results there. Sorry, here we go. I'm just pointing. This is how they're all connected there. Another way to look at this, and so this is a little bit different, but there's something called the block tree as a tool available at Family Tree DNA. This is actually something called the big tree that I gave as an example in your handout. But you can, I don't know if you can see this, but if you look for the Scottish flags over to the right, you can see that says Ogilvy, and that's sort of how we fit in comparison to other groups. I'm just gonna try to get through here really quickly. So that first match helped us get as far as Nova Scotia and then back to Georgia, right? That cousin, that sixth cousin. But we were kind of wondering about, like, it didn't really help us get any further back from there. But we did start to develop this genetic signature. So then an interesting thing happened. So when I was growing up, I heard that there was a family that still lived in Georgia that claimed to be descended from our Ogilvies. And I will tell you, when I was younger, I was kind of like, well, their name isn't spelled the same as ours, and they should just get their own ancestor, because clearly we're not related. Because their last name is Oglesby, which is different than Ogilvy, right? But I was able to connect through the wonders of the internet, was able to connect with a woman who had the same questions, same burning questions as I did. And she was able to convince her brother to participate in Y-DNA testing. And so we had this question of, is that person going to show up as a match to my dad and to his sixth cousin? So I can't see you all because of the format that we're using tonight, but I like to have people kind of vote on, who do you think for my dad, once my dad upgraded to the Y700 and got his, the terminal SNPs determined and got his new, not predicted, but known haplogroup, which one was he going to match? Was he going to match this person that had a haplogroup that was definitely an Ogilvy that had, closer Scottish origins, or was it going to match with this Oglesby relative, so-called relative that was from Georgia? And the exciting thing was my dad matched this relative from Georgia. So we were really excited to make that connection. And finally, we weren't able to say exactly how they're related, but they are definitely related on the paternal line. Okay. And so they are showing up. It's this blank one right here underneath that sixth cousin. And if you look over to the right, those purple 14s right there are those three testers, my dad, his sixth cousin, and this individual from Georgia. And there are other similarities if you look at the whole string of numbers and compare all of them. But we were pretty excited, and also very excited when we looked at the time predictor tool, because it suggested that where they branched off away from each other was right around 1750, the year 1750. And if you'll remember, that is very close to when Peter and John Ogilvy were both born. So we definitely feel like we're in the zone with these guys. There's more to be determined, but it was pretty exciting to make that connection. So we're solidly in Georgia. Just real quick, because I know we're getting short on time. Check the time real quick. I'm actually going to try to wrap this up pretty quickly, but I just wanted to highlight the power of surname projects. It's really, there's so many projects out there, and they're very variable. They can be geographic, they can be just surname, they can there's mitochondrial options. It's really exciting stuff. There's even, I didn't get to this, but on your handout, I mentioned that all of this Y-DNA stuff started because of interest in Thomas Jefferson. So I have the Jefferson project mentioned right there. And so this is just a screenshot of what our surname project looks like. And there's this really cool thing called a group time tree that is one of the newer features that I'll show you an example of. But this is where you can click into seeing these STR results, seeing everybody being compared there. And you can see I've got two different subgroups now of, you start being like that. One of these things is not like the other, and you start grouping, right? And then you can also look at SNPs, which I'm not going to delve into, but just there's a whole world out there that you can explore with SNPs. Okay, so this is the group time tree, and this is the really, really cool thing. And I actually called Family Tree DNA today just to make sure. But this basically helps you see those haplogroups and how they've split, and also helps you understand the time frame of when those splits were happening. And I'm just going to zoom in here a little bit. You can see here we have, it says to the right, James Ogilvy, born about 1725, and then James Oglesby. So that's my dad and his Oglesby cousin right there showing up. And they are both now under a new haplogroup that is right there. It says R-FTD89151. That is different than what I showed you just a few slides ago. That number that I showed you, if you follow it back, is right here. And so the thing that happened most recently that I'm so excited about is, out of the blue, we had a gentleman who lives in Scotland, and his family has been in Scotland. I've helped him research back at least until the 1700s, and likely much further back. And in this, basically the same parish for most of that time. It's a parish called Lintrothen. And this individual is showing up here just above my dad and his cousin. It says Thomas Ogilvy, and it says when his birth and death dates there. So what happened is, when he tested, Family Tree DNA reevaluated the haplogroups, and they kept him at that 88637 haplogroup, and split off my dad and his cousin and assigned them a new haplogroup. So it's almost like we got a new leaf even further out and down. So my dad and his cousin are downstream from this individual who lives in, his family has been in the same parish in Scotland for many, many years. I looked up, I didn't put the slide in, but I looked up their time predictor, and it said that probably they split off, and actually you can see a little bit on this chart here, but right around 1450. And so somewhere between that 1450 and 1750 is where my family split off from his family. Like that's the zone where I need to be looking for records. It'll only take me a little while. But we were incredibly excited to have that gentleman test because it changed our understanding, and it definitely gives us a specific location in Scotland to be looking at. Now they could have still come via Ireland, but we definitely have found our, a match there, and it only will give us, there's just, we will continue to build on this knowledge as more and more people continue to test. If you have questions, I highly recommend ISOG for more information. That's on your handout. This is about different DNA projects. And then more general information about Y DNA and about the Y chromosome. For testing strategies, I mentioned this at the beginning, but go big if you can, but it's okay to start at Y37 and keep upgrading as you're able. It's definitely okay, and it's actually recommended that you recruit people to test, to do some targeted strategic testing. So if you're female and you don't have a Y DNA chromosome or a Y chromosome, you can ask your brother, you can ask your uncle, you can ask your paternal line cousins to test, or representatives of whatever line it is that you're interested in. And some people have done that. They've been able to get multiple lines represented with various cousins. So, you just have to find the appropriate person that has the appropriate Y chromosome that you want to test. And I'll give you an example of this in a second, but you want to just think in terms of family representatives. With autosomal DNA, we're thinking test everyone, right? That's, people often say that, like, just hand out tests to everybody at the holidays. But in this case, you really only need sort of like a family representative. And you saw what I did there with my dad and his sixth cousin. I would say maybe if there's some questions about parentage, maybe you want to bring it back to like every 3 or 4 generations. But you don't need to have, each of your brothers, or if your brother tests, maybe you don't need your uncle, or maybe you have your uncle, so you don't need to ask your brother, that kind of thing. So that will offset maybe the cost of testing too. So just to help you visualize this, this is not my Ogilvy family. It's a different family, but the blue box going up is the direct paternal line. And this is a family where they daughtered out. The woman I was working with didn't have any brothers, and her dad was deceased. And I think also she didn't have any paternal uncles. So I was trying to help her find some representatives from the family to help her. So you have to go up and over and then down. So like in this case, there are 3 brothers, and you want to find out if any of them had descendants that are male that could test. And if you strike out there, then you have to go up another generation, and then over, and then down. So just some things to keep in mind as you're thinking about the testing. And I'll end here with this slide of resources. Again, this is in the handout, but I mentioned ISOG. There's so much available on YouTube. People have such wonderful presentations out there that you can access at any time. The Family Tree DNA has a learning center. So I think you need to have a test with them first before you can access it. There's Facebook groups. I think Roberta Estes, who's from Michigan, has some really great blog entries that you can check out. And then I'm a big fan of Legacy Family Tree webinars too. Some of their webinars are free, and some are only available through a paid subscription, but they're definitely worth checking out. So with that, I will go ahead and end. Thanks for hanging in on the wild ride of exploring Y-DNA.
Thank you, Ginger. Are there any questions? I think if you type a question, if you're on Facebook, we might be able to get something answered. But I did have one. Can you give the source of your testing again? Like, where can you test Y?
Yes, Family Tree DNA, and that's on this slide. The second entry here on this slide, Family Tree DNA is the company. And by the way, I don't get any, like, I'm not paid by them or anything to promote them or anything like that. That's just kind of the reality, is that that's where you go for Y-DNA testing.
Jessica, did you have questions?
No, I think she did a great job covering it. So thank you for doing that.
Yeah.
I will second the Legacy Family Tree webinars. They are a great resource. And I mean, I'm speaking as for myself, not as a representative of the society, but they do have a lot of really great videos on there about all sorts of genealogy related topics. So it's definitely worth looking at.
Mm-hmm. Definitely. Yes.
And I want to say just one more thing too. I loved the paternal line that you did and how you laid out that line of your family tree. That was kind of cool.
Like with the photos.
Yeah.
Yeah. I know. I love it too. Every time I see it, it brings a smile to my face. And I like to look at, like, okay, it looks like, these eyes or this balding pattern or whatever got passed on to a certain point. And then you can see, oh, that's where, this line starts to have an influence, or, you can see my grandpa is half Scandinavian. So you can start to see, his mom's, influence there. But yeah, I love it too, for sure.
Cool. So if you didn't get a handout, please contact me at Willard Library. You can give me a call at the library, and I can try to send it to you. And again, I apologize that I couldn't find an easy way to get it out through our presentation, but I will email it out if you want it. So I just saw someone was asking about a handout, so I just wanted to answer that.
Okay, for sure. Yep. And there aren't many Ginger Ogilvies out there. So if you just, search for my name, I'll come up, and you should be able to, grab my email address too. Or I can make sure Jessica has a copy of it too. Mm-hmm. If anyone needs a copy, I'm happy to pass that on or ask or answer any questions. Sometimes with DNA, people may not feel comfortable asking questions in a group setting, and I'm always happy to answer questions privately. So
thank you.
Yeah.
And Jessica has it, and I have your contact information as well, so we can help with that too. So.
All right. Well, thanks for the opportunity. I'm going to go ahead and stop sharing here.
Yep. Thank you. And I can do that.
Oh, okay.
Great. Sorry. So thank you very much, Ginger. And thank you, Jessica, for joining in and helping with the program. So
of course,
I think we can sign up. Yes.
Right.
Thank you.
Thank you so much, Ginger.
Yeah.
Appreciate it.
Bye.
My pleasure. Bye.
Good night.